Okay, hey, let's see. I think I'm in. Perfect. Hey, DNA today again. More of the same. I'm pulling up this slide again to kind of emphasize a point here. Yesterday we spent a lot of time talking about the combinations of adenine, thymine, cytosine, guanine, and the particular combination, the whatever combination of those four letters that you have in a particular chunk on your genes essentially determines your traits. Now we're going to find out that there's some on and off switches in there. There's some ways they get expressed and don't express, but those are the instructions to the human body. A very interesting thing that's going on right now that's worth pointing out is we are starting to develop technology to read these pieces of code. All right. I'm sure you've heard of 23andMe, Ancestry.com, those kind of things. Those are companies that are taking DNA samples and they are pulling them apart and they are quote unquote reading your DNA. What they're looking for is they're looking for particular chunks of A's, T's, G's, and C's in particular areas that tend to correlate to certain heritage spots, certain genetic disorders and diseases. And so it's an interesting study here. I have a video that is in the notes here. It's old. Okay, it's from maybe 15 years ago. And it's talking about this same technology, only back then it was $1,000. Now I think 23andMe is like $50. You can get a test, and it's worth checking the video out, at least the first part there. But it is something worth pointing out. Your generation, okay, the younger people out there, you will probably live in a time where you'll be able to read your genetic code. Okay, we're already dabbling in that area. You'll probably be able to get your DNA tested and know your likelihood of having certain diseases, whether it's breast cancer or Alzheimer's or anywhere in between those two. Genetically inherited diseases, you'll probably be able to go in and determine a pretty precise likelihood of if you're going to have those or not. The technology will soon exist that you'll be able to DNA scan your children. Now, we can already do this looking for genetic disorders, but we might be able to go in there and see, hey, you're going to have a child with this heart condition. They're going to have these particular traits. And you'll probably be able to do that before giving birth to that child. Okay, so it'll be some interesting moral dilemmas in our future as far as trying to determine what is right, what is wrong, those kind of things. And then the last one is maybe a little bit further away, but still, as we're looking at those chunks of code, hypothetically and now practically, thanks to technologies like CRISPR, we are able to cut and paste certain genes into and out of your DNA. Now, it's still a very rudimentary process, but the tools are being developed right now to change your DNA. So interesting things coming in our future of genetics. Like I said at the start of the unit, we're just scratching the tip of the iceberg on DNA. We just figured out what it was, and we're still figuring out exactly how it works. Where's DNA located? We know that DNA is located in the nucleus. Remember, though, we also find it in the mitochondria. Side note, I was just talking about Ancestry.com and 23andMe. They use maternal DNA or mitochondrial DNA to determine your heritage. Your mitochondrial DNA, if you remember, you get from your mother. And so it's not combining every generation. It's relatively unchanged as it gets passed down. So it's a lot easier. It's a lot more preserved. And so it's a lot easier to trace back to, 10,000 years ago, or whatever it is there. There's still some legitimacy problems with that, 23andMe stuff there. It's a ballpark is kind of what it does for you. Hey, today, the main takeaway today is this DNA replication process. We know, because we just got done talking about mitosis, that your DNA is going to make copies of itself. it is the instructions. And so as you make, more copies, you should probably include the instructions in there as you're going. It's kind of an interesting replication process. If you look here, we're just, guys, we are just going to scratch the surface of it. There's a ton of different moving pieces and parts and enzymes. And if you take like an AP biology class or college bio, you'll go through exactly how this all works. What we need to know is when we have an old strand of DNA, there's going to be an enzyme that unzips the DNA, right? It unspools it and unzips it. And then new bases are added to there, new nucleotides are added in. And so what you get is you get two strands of DNA that are each half old, half new. So kind of an interesting look there. We call that semi-conservative. So it's not like, if you're making copies on a copier, you have an original and then you have a brand new copy. DNA doesn't work like that. It splits the original in half. So it's half and half as you're going there. So in theory, there should be a little bit less chance for mutation that way. But that is how it works. A's bonds with T's, G's bonds with C's, same old, knowing that it is semi-conservative as it goes. And there's a lot of checks along the way to make sure there aren't any errors. Here's an interesting video that's worth checking out of what it molecularly looks like inside of a cell. Once again, a process that is it's not too complicated. It's more complicated than we're going to talk about. And then a video there too, if you want to check it out. This DNA replication thing, it's actually why you age. You have these things on the end of your DNA called telomeres, and they're like extra chunks of DNA. And every time you copy your DNA, you can't quite copy the whole strand. You leave the ends out. And so your DNA gets shorter each copy. Thankfully, you have these telomeres that are nothing. They just make room for copies. But at some point you run out of them, and then that DNA can no longer replicate anymore. That is where aging comes in. And so if you could regenerate those telomeres, hypothetically, your DNA would not age. Wouldn't that be weird? Okay. Today we are going to be talking about transitioning into our next deal here, and that is RNA. Okay, RNA is DNA's half brother, I like to call it, because it is half of DNA. Okay, so RNA is the messenger. DNA stays inside the nucleus. It does not leave. Instead, it relies on RNA to venture out and do things for it. RNA is a single strand. That's why I call it its half brother. It's only a single strand. It contains a ribose instead of deoxyribose. And instead of T's, we're going to be using U's instead. So in the base pair below, the A and DNA is going to bond. Oh, shoot. I thought I had it written in there. A bonds with U, T bonds with A bonds with U, and then G's and C's are the same. So essentially for RNA, whenever you want to put a T down, put a U instead. This is where, and we're going to spend the next 2 days talking about this. This is where it gets the function of it. Okay, what it's actually doing. So DNA are your instructions, right? RNA is actually going to be the one that takes the code on that gene over to the protein factory, the ribosome. All right, so we're going to spend 2 days talking about that process where RNA takes the information from the gene, delivers it to the protein factory. The protein factory pumps out whatever cells it needs to in order to express that gene. All right. And I'm going to leave you with, there it is. Cool. I'm going to leave you with this. Yeah, right here. Okay. Write this down. This is, actually they call this the central dogma of biology, which is a big, interesting term to use there. But this idea that DNA is going to be converted into RNA, and then it is going to be used to build a protein, and proteins are what build you and I. All right. As we move away from cells and cell parts, this is going to be huge. Okay. This is how genetics works. This is how evolution works. This is how traits work. This is essentially, essentially it's called the central dogma of biology because this is how biology works. All right. You take DNA, it is going to be converted into RNA, and then that is going to build a protein chain from there. All right, so I'm going to stop there. I feel like that's enough information for today. We'll dive into transcription translation next time I see you.